Hey, Hammy here. Coming today with a video on 7.3 covering mutations. What happens when the DNA gets screwed up and what effects does that have? Okay, a mutation is any change that happens in a sequence of DNA or RNA, because remember, they're eventually going to lead to the formation of a protein. Okay, so down here on our DNA molecule, when one of those bases, A, T, C, or G, gets screwed up, then it's going to, lead to something different in the protein possibly, and we call that a mutation or a change. Okay, sometimes they happen spontaneously during DNA replication when DNA is being copied, or when it's being transcribed into mRNA. Sometimes it messes up and plugs in the wrong base. Sometimes it's something in the environment. Okay, when we call that a mutagen, when something outside the cell affects the DNA inside of it. Okay, some examples of mutagens, for example, and these are kind of A, B, C here in your notes. A for radiation. You can write down things like UV radiation. Okay, the ultraviolet light from the sun, okay, penetrates our cells and can screw up the DNA and lead to things like melanomas and stuff in our skin. X-rays would be another example. If you've ever had X-rays done, you notice that they put this big lead vest on your chest, or they cover up parts that don't need X-rayed, and then they leave the room. Okay, why is that? Well, the X-rays, they're helpful, okay, to diagnose like a broken bone, okay, but you don't want to be overexposed to them. Think of the radiologist; they're taking X-rays all day long. So if they wouldn't leave the room and they'd get exposed to these things all the time, okay, that could increase their risk of dangerous mutations and cancers and that kind of stuff. Just getting an X-ray every now and then at the dentist or at the ER for a broken bone is more helpful. It kind of outweighs the risks that come with that. Just a short one-time kind of thing. Second one, chemicals. An obvious one is cigarette smoke that you hear a lot about. Okay, there is dozens of different carcinogenic, mutagenic chemicals in that smoke. Carcinogenic, meaning causing cancer, and just basic, that would fall under mutagens too, that cause changes in the DNA. One you hear about in preserved meats, so hot dogs, pepperonis, different kind of beef jerkies, they put nitrates in them. Processed meats, it's also in wine to help preserve the wine. Okay, so in some types, those can be harmful. I've heard barbecuing before. Actually, barbecuing, the soot or smoke that's given off can actually be small mutagens. But again, nothing to be fearful of. Just, once in a while, not overdoing it. Benzoyl peroxide, some acne products, you have to be careful about what ingredients they contain as well, and often how often you're using them. Sometimes they're beneficial, but you shouldn't be overusing them. And the last thing, sometimes infectious agents like viruses or bacteria can cause damage to our DNA. One you hear about in the news recently here is HPV, that they do provide a vaccine for now. It's a sexually transmitted virus, but they have found that it really increases the risk for cervical cancer in women. And then some other throat cancers and stuff like that can happen in women and men. And so, they suggest getting a vaccine for this to prevent those types, getting the virus, which then could cause those types of cancers. Helicobacter pylori is another one that can mess up your digestive system. Sometimes they check if someone has chronic acid reflux or GERD. They'll check to see if these are in your stomach, if there's over acid production. And they can mess up sometimes the DNA and stuff in your intestinal system. So just some examples of some mutagens that can kind of screw up your DNA in your cells. Two major categories of mutations are germline and somatic. Okay, germline doesn't have anything to do with like germs, like getting sick. Germ meaning like in an egg or sperm, or in that embryo or zygote. Okay, in those initial stages. Okay, so example, if a parent has a mutation in an egg or sperm, and that is the one that is fertilized and becomes, grows into the embryo of the new child, that is going to be passed on. It can be inherited. Or if it happens in the embryo of the child, when you were first that little ball or clump of cells, okay, all of your cells are going to have it, okay, and it's going to affect your whole body. Whereas somatic often refers to body cells, so in certain tissues. For example, if a woman develops breast cancer, it's in that tissue, and it's not going to be inheritable. It's only going to affect that person, and then again, it needs to be treated in various ways. But it's just in other cells in the body. And so this very much affects kind of whether they're passed on and how they're treated and how they affect future generations. Okay, now to talk about some of the specific types. One of the first, I guess, bigger categories of specific types, if that makes sense, is chromosomal mutations. When pieces of your chromosome break off and rejoin incorrectly. Okay, so remember DNA makes up that chromosome. So if the chromosome is getting messed up, that in turn is messing up your DNA. Results are very serious, often result in the death of an organism if it happens in parts of chromosomes. If it does, it forms a serious genetic disorder. One example that is viable is Down syndrome, where there's an extra 21st chromosome. Okay, so obviously these individuals live, but then it affects kind of how the body can function. And a lot of us are familiar with someone with Down syndrome. How can these mutations happen? Well, there's 5 different types. Okay, a deletion is the loss of a part of a chromosome. So here you see the normal chromosome, and then oftentimes during crossing over, during meiosis, remember how these things cross over, switch, and break? Maybe they don't break equally. And so these two can often kind of go together. During meiosis, one might get an extra piece, whereas the other piece that lost it, okay, this was supposed to go here, ends up switching to the other one. So one will have a duplication, an extra piece, and one of the homologous chromosomes will have a missing piece. An inversion is like during crossing over, if it gets put upside down. So you see wide, thin, thin. Over here, it's thin, thin, wide, if you look at the banding pattern. Okay, so it got switched. This creates problems for reading that gene, for transcription and stuff. When they need to make, if those genes on there, it needs to go in and read backwards, and that can be tricky for the cell. And sometimes you can't get enough gene product, or it doesn't work right. 4th type and 5th type kind of go together, insertion, and that's where you get a gene break off of one chromosome, and translocation, it ends up going to a non-homologous chromosome. We call these we call them jumping genes or transposons. Okay, and one of the interesting transposons in the human genome is the ALU sequence. Okay, the ALU sequence we find over 1 million times in the human genome. Okay, and this thing has been able to replicate, and we find it all throughout the human genome system. And problems with this can often lead to certain genetic disorders. Okay, but they different chromosome that they're not even supposed to be on, or they were not on originally. A second more specific type is a point mutation. Okay, just like you think of one point, we're talking point like as in one base pair on the DNA, a single nucleotide. So an A, T, G, or C gets made wrong. Okay, not a whole big piece of a chromosome, which could be thousands of base pairs. We're talking one base pair. These tend to be less serious, obviously, because there is you're talking less DNA. There's 3 types of point mutations: silent. If you remember back when we looked at the genetic code chart, sometimes, let's say, this A right here gets changed to a G. Well, if you look at the chart, CAA is glutamine, CAG is also glutamine. So remember, some amino acids have more than one codon that codes for them. So if you have a mutation that codes for the same amino acid, what effect is it going to have? None. It's silent. Yes, there was a mutation, but it makes the exact same amino acid, so the protein will still function normally. A missense is when it changes it to a different amino acid. So let's say the second nucleotide here in this protein, CAA, goes to CCA. Now you go from glutamine to proline. Okay. How is this going to work? Well, it just depends. You could have a gain of function, okay, where it might change that protein and give it a slightly different function, or you could get what we call a loss of function. A loss of function means now this new amino acid in that string of however many amino acids is in that protein, this might change the protein enough that it does not work. Okay, and so sometimes it won't affect it a whole lot. It might just change it a little bit, or might do something different. Other times it won't work. Okay, and that loss of function can also go with the nonsense mutation, where here UAA or CAA gets changed to UAA, which is now a stop. Okay, so now you're reading along, and let's say you're supposed to have a whole bunch of amino acids after that. Now you have a stop. That's fairly serious. You're going to have a loss of function. That protein is not going to be the same, is not going to work the same. Besides the substitution, okay, which we were looking at in the last slide, a point mutation, which is just one subs in for the other, you can also get a deletion or an insertion. So you can take out one and have a missing one, or you can add an extra one. These cause frameshift mutations. Okay, so normally the reading frame, we say, is a triplet, okay, 3 at a time. Every 3, so DNA, remember, gets changed to mRNA, and then that gets translated to a protein. Okay, if you have a change in the DNA or the RNA, okay, for example, here they're showing either on the DNA or the RNA, we lost a nucleotide. So they're taking this U out. Well, what's that do? The beginning is going to be the same, methionine, lysine, but now instead of UUU, which codes for phenylalanine, now you take that out, that shifts everything over. So frame shift, reading, changing the reading frame. Now it's going to be UUG, which is leucine, which is not phenylalanine, and it's going to change everything after that. So now instead of glycine, you have alanine, and instead of isoleucine, now you have a stop, which is, again, pretty serious, because instead of continuing on and making the rest of the amino acids, it's going to stop, and you're going to have a non-functioning protein. So frameshift mutations are usually pretty serious and pretty bad. Finally, when we look at kind of some overall effects of mutations, it's important, before you get too nervous, that a lot of mutations are just neutral. Okay, they're like the ones where we looked at where you subbed in a base and it made the exact same amino acid. Okay, so it's not going to have any effect. Sometimes, your DNA polymerase will come along and it'll repair the mutation before it is propagated any further. Sometimes, remember, you have exons and introns. Sometimes maybe the mutation might happen in an intron or part of your DNA that is not coded for any type of protein, so it will not affect anything. Sometimes there are beneficial mutations which have a positive effect. We often call beneficial mutations the raw material for evolution to occur. Okay, in other words, how do things change? Well, their DNA has to change. Okay, so for this idea of adapting to changes in the environment, Well, their DNA has to change. So sometimes beneficial mutations might change the phenotype of an organism for the and lead to what we'll get into in future units called natural selection. It will help them to survive better and pass on that mutation. And finally, what we often think of as harmful when the results is a protein that does not work. This is a main cause of genetic disorders. Or in the next section of the chapter here, we'll take a closer look at cancer and how mutations can affect our normal body cells and make them go out of control. Okay, so remember that mutations aren't always bad. Sometimes they're not noticed, they're neutral, and sometimes they have positive effects.